Hello, everyone. My name is Jessica Miesel, and I'm an assistant professor at Michigan State University. My research focuses on understanding the characteristics of charcoal-like materials and how they influence ecosystem processes, such as nutrient cycling and nutrient availability in soils. I study natural chars produced in wildfires and prescribed burns, as well as biochars, or human-created chars that are used as soil amendments. And I want to thank Dr. David Overstreet and Dr. Frank Kutka for the opportunity to present to you today about what modern biochar research can tell us about charcoal in ancient gardens. In my presentation today, I will provide an introduction to biochar and describe the current understanding of how biochar influences soil fertility and plant growth. And this information may help provide insights about how and why charcoal was used as amendments in ancient gardens. But before I begin, I want to point out the image shown on the left side of this first slide. This is a scanning electron microscope image. It's a very high magnification, and it shows a highly magnified photo of charcoal produced from pine wood. And the structures you see here are the plant cell walls. And you might even be able to see some of these pores, which are the pores that connect the walls, the cells. And this physical structure stays intact even after wood is charred. And this physical structure is an important part of how and why biochar can influence the physical, chemical, and biological characteristics of soils. So I will discuss some of these topics in more detail later in the presentation. But I want to start with what biochar is actually. First, biochar is a type of charcoal-like material that is produced from biomass, and usually plant biomass, and it is applied to soil for a management purpose. So the human intention is an important part of this definition of biochar. Not all charcoal-like materials are considered biochar. It is only considered to be biochar when it is intentionally applied to soil. And charring happens when biomass is heated without oxygen present. This can occur either in a closed container that limits how much oxygen is available, or in a pit or mound that's covered with soil or other types of debris. And biochar typically has a very high carbon content. This is one of its defining characteristics. And it's also very low in nutrients, so it's not really a fertilizer source. For example, wood in general is about 50% carbon by mass compared to biochar, which can be up to nearly 95% carbon by mass. So biochar is much more concentrated in carbon atoms than the biomass it was produced from. And this is one of the most important characteristics that influences its ability to survive in soils for hundreds to thousands of years. When it is mixed into soil, biochar can influence the physical characteristics, such as bulk density, structure, and water holding capacity. And it also influences the soil chemical characteristics, such as pH and nutrient availability, and it can influence the biological communities that live in soil, which are so important in maintaining soil health, soil fertility, plant growth, and crop yields. So all of these characteristics have led to biochars being promoted as sustainable soil amendments, not because they provide nutrients, which they don't really do, at least very much, but because they influence soil characteristics in ways that can help improve crop yields. So although research on biochar is still relatively recent, biochar is not really a new technology. In fact, research interest in biochar began in the Amazon region in areas described as terra preta or dark earth soils. These are considered anthrosols from anthropogenic, human influenced. These are, which means these are soils with unique characteristics that were created by human activity. These terra preta soils occur near ancient village sites dating from about 1,000 to 9,000 years ago. And in tropical regions, such as the areas where these terra preta soils were discovered, tropical soils are very old and they're very weathered, and they're very well known to have short lifespans in terms of their ability to support agricultural productivity, even with fertilization. So an example of this is shown in this set of photos on the figure. And on the left, we see like a characteristic tropical soil. showing very weathered soil characteristics and really poor maize growth in the upper photo. But terra preta soils are unique because they have very high productivity and the productivity is sustained. Some of these sites have been cultivated for decades and don't even require supplemental fertilization. You can see a photo here on the right of a terra preta soil. And this is showing that the soil is very dark from a very high concentration of organic matter, including charcoal. And these terra preta soils support very strong maize growth and yield, which you can see in the upper panel here. So when researchers began investigating why these terra preta soils had such high nutrient content, they discovered high concentrations of charcoal, ranging from about 0.25 metric tons to 3.5 metric tons of charcoal per hectare. They also found that up to 35% of the organic matter in soil was present as charcoal. And in addition to a lot of charcoal, these sites also have other signs of human activity, such as fish and other animal bones and pottery shards. So these sites are understood to be the kitchen middens for these ancient communities. Basically, the discard piles for cooking fires, which became important garden sites with benefits that are still evident today. And discovering that charcoal was such an important component of the terra preta soils is what led to research interest in biochar. The basic question is, if ancient charcoal materials have such long-lasting positive effects on crop growth, could we improve agricultural productivity today by adding charcoal to soils? And could these modern biochar amendments help decrease the need for synthetic fertilizers and decrease the need to irrigate? All of these benefits have potential to help create more sustainable agricultural systems. So Dr. Overstreet asked me to talk about what current biochar research can tell us about how and why charcoal was used in these ancient gardens in the Menominee Nation. And I think it's important to acknowledge that what we know about biochar is really rooted in the agricultural practices of pre-Columbian cultures in another part of the world. So biochar is charcoal that is added to soil to achieve a management purpose. But what exactly is charcoal and how is it formed? Charcoal or charred biomass is thermally decomposed biomass. This is called pyrolysis, the breaking of chemical bonds caused by heat in the absence of oxygen. But pyrolysis is also the first phase of the combustion process, and it also happens continuously during a fire. So you'll have pyrolysis, which is heating without oxygen, and flaming combustion, which happens when oxygen is present, both happening at the same time in different parts of a piece of fuel and in different parts of a fire. This diagram here uses wood as an example fuel. The first part of the diagram shows pyrolysis happening, heat without oxygen, which creates char. This is an endothermic reaction, meaning it requires heat for the pyrolysis reaction to happen. Pyrolysis creates char, but it also produces some gases. These gases ignite when they come into contact with oxygen, and that is what produces the flames we see in a fire, oxidation of gases. This is an exothermic reaction, which means it produces heat, and that's a heat we feel from a fire. So as the gases continue to combust, more and more of the carbon in the biomass will be released. As long as oxygen remains available, ultimately, a piece of fuel will get converted to gases, leaving mineral ash or white ash behind. This white ash is the non-combustible part of plant biomass. In reality, such as during a cooking fire or campfire, pyrolysis and combustion are both happening at the same time. And when the fire is put out, you will have a mixture of black ash or charcoal, which still contains a lot of carbon, and white ash, which is the mineral residue of plant biomass that has very low carbon content. So how much black ash or charcoal versus how much white or mineral ash is left depends on the characteristics of the fire. This diagram shows the temperatures at which the elements or nutrients volatilize or convert to gases during a fire. The lower color bar here shows the degrees in Celsius. And since most cooking fires or forest fires have oxygen present, we'll start by looking at this band here for ash characteristics. This shows that at around 150 degrees Celsius, biomass starts darkening. This is the beginning of the charring process. Char or black ash begins to form around 250 degrees Celsius or to 400 degrees Celsius. And between 400 to 600 degrees Celsius, we tend to get more white ash, the minerals that are left over after the carbon has been combusted. The diagram also shows that nitrogen volatilizes between 200 to 500 degrees Celsius. If we connect this to the ash band, we can see that the black ash will contain more nitrogen than white ash because white ash is the residue from higher temperature fire, meaning more of the nitrogen is likely to be lost. All these other nutrients, potassium, phosphorus, magnesium, calcium, and others, volatilize only at much higher temperatures. This means that if a fire is not hot enough to volatilize these nutrients, they will still be present in the white ash left behind from the fire. So that is how heat influences the chemical composition of fire residues when oxygen is present. But if oxygen is not present or is extremely limited, we can look at this black carbon continuum bar. And black carbon is just one of the many names for charcoal-like materials. So we can call it black ash, black carbon, pyrogenic carbon, charcoal, or char. There's a lot of different names that we use to describe the material. Charcoal or oxygen can be limited. For example, if a fire is started in a container that is then sealed, or if fire is started in a pit and then covered with soil or another type of material that would limit oxygen and not burn up. This black carbon bar shows that temperature influences the extent of charring, with low heating temperature producing only slightly charred biomass, ranging up to charcoal at higher temperatures, and ultimately soot, which is nearly totally carbon in a very concentrated physical and molecular structure. Biochar production systems can range from very simple systems, like a campfire, to very high-tech systems, such as this room-size industrial system. This upper row of photos shows some of the ways that it can be produced as part, that charcoal, biochar, can be produced as part of a non-industrial culture. This ranges from cooking fires here to slash and burn agricultural systems that create or expand areas for gardens. This third photo here shows a relatively recent management fire in the Apostle Islands used for cultural value to increase blueberry production. And although this fire that's shown in the photo was conducted with some modern prescribed fire tools, we know that Native Americans across North America used fire to manage the landscape long before Europeans arrived. All of these activities would create char and ash that would eventually become mixed into the soil, whether or not it was intended as a soil amendment. The fourth photo on the right, on the top here, shows an earth mound kiln system that is from Kenya, and it's used to create charcoal. Then if we move to this lower row of photos, this shows the range of more industrial processes that are used today to create biochar or pyrolysis products. This ranges from low-tech cooking stoves designed to just increase efficiency of fuel wood, to backyard kilns, to portable or large-scale pyrolysis systems. And the higher tech systems allow the operator to control the pyrolysis conditions with more detail to be able to create chars with consistent characteristics. And some of these higher tech systems can produce bioenergy that either heats or powers a facility, and they produce charcoal residues as a waste product, which that can then be used as a soil amendment. So the connection between bioenergy systems and biochar is one of the reasons there has been so much research interest in biochar today, because there's potential to fine-tune bioenergy systems so that we can decrease use of fossil fuels while also producing a byproduct that has agricultural value. Dr. David Laird with the USDA calls this a win-win-win scenario with benefits for bioenergy, for economics, and for the environment. We've already touched on one of the unique characteristics of biochar, that it is highly concentrated in carbon. The proposal for combining bioenergy and biochar production is that pyrolyzing biomass would convert about half of the carbon to energy, while the other half becomes stabilized in biochar, which is then returned to the soil. So instead of releasing biomass carbon into the atmosphere, either through natural decomposition or by burning for energy, creating biochar as part of a bioenergy system returns about half of the biomass carbon to soil, keeping it out of the atmosphere. This is why you may hear biochar being described as a climate change mitigation strategy. And this is because it helps keep some of the carbon out of the carbon cycle. And when added to soil, it can increase plant growth, enabling more carbon to be taken out of the atmosphere by plants. So I hope that all this information helps understand what biochar is, how it can be created by natural and industrial systems, and why it has a potential role in improving environmental sustainability beyond just as an agricultural amendment. So now I'm going to talk a bit more about biochar itself. One of the things that I think is so fascinating about biochar is that the charring or pyrolysis process changes the molecular structure of biomass, but leaves the physical structure mostly intact. You can see in this photo a tablet of compressed grass or hay. This entire tablet can be charred, and when the tablet is crushed, we can see that the physical shapes and size of the fragments of grass leaves and stems still exist. When we zoom in to very high magnification, using a scanning electron microscope, we can see the physical structure of the plant cell walls is also still intact. The scanning electron microscope images on the right are from charred pine wood. So again, you can see the shape and size of the plant cell walls and the pores that connect the cells. If we zoom in another 10,000 times closer to this image, zooming in as shown as this image number 2, we can see these areas with linear patterns highlighted in this yellow box. And we'll call these organized domains. And this is a transmission electron micrograph, which is 10,000 times more magnified than the SEM image on the right. So these organized domains, shown by this linear pattern, occur when the charring is extensive enough to create a crystalline structure in the molecules, which is seen even closer in figure 3. And this consists of many, many rings of carbon atoms connected in what is called a polyaromatic ring molecular structure, with each individual aromatic ring made up of 6 carbon atoms connected to each other, and each of these rings is connected to many other rings. This produces a honeycomb pattern of carbon atoms, which we can see here, where each corner of each of these rings represents the location of one carbon atom. So you can see a very dense structure of carbon atoms. So this molecular structure does not leave room for many other types of atoms or elements or nutrients to be present in the biochar, which is why biochar is high in carbon and tends to be very low in other nutrients. But you'll notice that this image number 2 up here shows an organized domain as well as a surrounding area that we'll call a disorganized domain, or areas that do not have this linear pattern. And that is because the area with the linear pattern represents areas with the most condensed molecular structure, or the area most concentrated with carbon present as these 6 carbon ring structures. But the areas outside of that linear pattern, or the disorganized domains, are not quite so concentrated in 6-carbon rings. So in any piece of charcoal or any piece of biochar, there will be some areas that are very concentrated in 6-carbon rings, such as shown here in the circle on the left, as well as other areas slightly less concentrated or less dense in carbon atoms, shown in the oval on the right. So any level of charring will create 6-carbon rings because pyrolysis, by definition, breaks chemical bonds with heat, essentially rearranging carbon into these 6-carbon ring structures. So it rearranges the molecular structure of the original biomass. So if you think back to the diagram of temperature thresholds, in general, chars formed at higher temperatures will be more concentrated in these 6-carbon rings, and chars formed at lower temperatures will be less concentrated in the 6-carbon rings, or have a greater variety of elements and chemical bonds present. These other areas, you can see there's oxygen, an OH group. These other areas contain atoms or elements other than carbon. So these create reactive sites, which typically have a small positive or negative electrostatic charge. And these charged sites you can think of as kind of like sticky ends on the molecule that help it interact with or bind to other molecules. And this can help it attract to molecules such as water, parts of soil minerals, or even other types of organic matter. And these reactive sites are really a big influence on how biochars or other charcoal materials interact with soil. So what can biochar research tell us about how charcoal amendments were used in ancient gardens? In the next few slides, I'll show you some examples of how biochar can influence the chemical, physical, and biological characteristics of soil. And although we can study each part, it's important to remember that all 3 of these characteristics interact with each other all the time in soil, and together they influence soil health and soil fertility. So understanding how biochar affects each of these parts can help us identify insights on why biochar was used in ancient cultures. So I'll start with focusing on this question here, why biochar may have been used in ancient cultures, by digging into what we know from the current biochar research. So I'll start with some examples of how biochars influence soil chemistry. This graph on the left here shows mineral soil pH measured in a field scale study of pine wood biochar at Michigan State University. And these measurements were taken 2 years after adding biochar to the soil. And I'm showing 3 treatments here. The no biochar control shown in this pink color, and a low level of biochar, which is about 11 tons per acre, and also a high level of biochar, about 33 tons per acre. The data for each treatment are shown in these box and whisker diagrams. So for each treatment, the median or the middle value is shown by the dark horizontal line across the inside of the box. The length of the box represents approximately the middle 50% of the data, and the whiskers or the vertical lines show the range from the minimum to the maximum pH value measured in each treatment. And the black dots here are outliers or extreme values that were very different from the rest of the data. So this figure shows us that biochar, low and high levels, increase soil pH, and the more biochar we add, the higher the pH. So these measurements were taken 2 years after we added the biochar. So we can consider this to be a relatively persistent effect of biochar on soil pH. And changes in pH in soil are important because pH influences how available soil nutrients are for plants. And even if there is no change in the total amount of nutrients in soil, pH can determine how well plants can actually access those nutrients and use them for plant growth. And this diagram on the right shows how nutrient availability changes with pH for several nutrients that are important for plant growth. So the pH scale is shown here on the horizontal axis on the bottom. And for each nutrient, the thickness of the bar corresponds to its relative availability in soil. So it's availability for plants. So you'll see that for nitrogen through magnesium, they're most available in this middle range of pH values, around pH 7, which is neutral. And you'll see in the box plot diagram that the pH in our no biochar control treatment centered around pH 7. So nitrogen, for example, up here on the top, is most available in the middle range of pH values, and its availability decreases if at lower pH values, and also it decreases at higher pH values. So this is important for understanding why charcoal may have been added to soil in the ancient garden sites, because soils in the northern parts of the Great Lakes region are often very sandy and tend to be on the relatively acidic side. And although I don't know what the pH is at the ancient garden sites, I have other research sites in sandy soils in Michigan and in Wisconsin where the average pH value ranges between 4.5 to 5. And you'll see at pH 4.5, the nutrient bars are relatively narrow, which means even though there may be nitrogen in the soil, it will be difficult for plants to access that nitrogen. So this means that if you're trying to grow food crops in acidic soils, the plant growth may not be very strong because they just can't access the nutrients that are in soil. This can sometimes lead to nutrient imbalance, and plant growth will not be very strong. But by adding biochar to soil, especially on the soil that's relatively acidic, it may be possible to increase soil pH in ways that then improves plant ability to access nutrients from the soil. So you can see this here, that the bar thickness for nitrogen increases if you move from pH 4.5 to 5, or from 5 to 5.5. So biochar can change soil chemistry in ways that increases nutrient availability for plants. Another way that biochar can increase nutrient availability is by helping to hold nutrients in the soil profile, preventing them from leaching out of the soil when it rains. This can be especially important in areas or in sandy soils that we know are common across the northern Lake States region, Michigan, Minnesota, and Wisconsin. Sandy soils are well known to have very low capacity to hold both water and nutrients. And these close-up photos here and the diagrams show sand and silt-sized particles. This helps us understand why sandy soils are not very good at holding water and nutrients. The sandy soils are basically like a sieve with these huge spaces between the sand grains, so water and nutrients just run right through. In contrast, if we look down at the diagram of the silt-sized particles, we see that soils that have smaller particles like silt and clay, they tend to be better able to hold organic matter, nutrients, and moisture. And then this helps provide a better growing environment for plants. So the graph on the right shows some data from one of my biochar studies where we measured how much nitrogen is leached out of a sandy soil during simulated rainfall. And we used 5 different levels of biochar here. And the levels range from a no biochar control, shown in this white bar by the white color, to 100 metric tons per hectare, and shown in the dark bar, the black bar, with the amount of biochar corresponding to the shade of from white to black. The darkest bars are really pretty extreme levels of biochar amendments. And this is much higher than would be realistic to add in a modern agricultural situation. But part of our goal in this study was to try to identify if we added too much biochar, whether it would create negative effects. But in this figure here, I'm just showing our leaching data. So the height of the bars shows the average value for each treatment in terms of how much nitrogen was lost from our system across the 2 years of our study. This is a total amount of nitrogen lost. And these data show that biochar level, the more biochar we add, the better we are at keeping nitrogen in the soil. And again, we see the more we add, the stronger the effect of biochar. So this and the data I showed you recently provide 2 examples of how biochar can increase nutrient availability for plants growing in sandy soils. First, by increasing soil pH, which helps plants access the nutrients that are in the soil. And second, here, by preventing nutrient leaching out of the soil from the soil profile. This helps hold nutrients in the soil for a longer time, helping plants access them over a longer period of time. Oops. So you might be wondering, how and why does biochar prevent nutrients from leaching out of soil? How does it help hold nutrients in the soil profile? In this sandy soil diagram here, I've shown biochar particles mixed into the soil. So I've shown them with little pointy edges just to illustrate the reactive sites I showed earlier in the diagram of the molecular composition. These are the parts of the biochar molecules that can react with or bind to other molecules, such as the molecules of essential plant nutrients, such as nitrate, ammonium, phosphate, calcium, magnesium, and potassium, all of which are essential plant nutrients. And each one of those essential plant nutrients, when they are present in soil, they all carry a small electrostatic charge. So this means that plant nutrients stick to these reactive sites on biochar particles, helping the nutrients to stay in the soil profile longer. And this diagram also shows that the biochar particles can help physically fill in the spaces in between sand particles. So this is one way to add organic matter to soil, to sandy soils, to help hold moisture into the soil. So, and that takes us now to looking at how biochar can change the physical characteristics of soil, such as moisture content, even soil structure and bulk density. And biochar is very porous, mostly because the plant cell walls are still present, like we saw in some of the SEM images that showed the plant cell walls and the pores that connect the cell walls. But even though the three-dimensional cellular structure of the original plant biomass is still present after the biomass is charred, the biochar can also be very physically fragile. So it can get broken down into smaller and smaller pieces over time as it ages, getting worked into these big spaces into grains of sand. So both by being able to absorb water into its pores in the biochar and by filling in physically in between sand particles, biochar can increase soil moisture content. This is another effect that is especially important in sandy soils, and it enables plants to have better access to the moisture they need for growth. The graph on the right is from the same biochar study I just mentioned, but this time I'm showing biochar amendment level on the horizontal axis, ranging from no biochar to our very extreme 100 metric tons per hectare level. I'm also showing volumetric soil moisture content on the vertical axis. And the different colored lines here represent the different combinations of soil texture and tree species that I planted in the treatments. And this figure shows us that soil moisture increases with biochar level, with the volumetric moisture content approximately 10% greatest at the 10% greater at the higher level of biochar amendment compared to the no biochar control. So this is important because increasing a soil's ability to retain moisture means that plants can make better use of rainfall and better use of any irrigation that happens. And again, this is especially important in sandy soils, which naturally have very low water holding capacity. So over time, biochar can also change the abundance and size of soil aggregates and can even change soil bulk density. And these types of changes also can help improve conditions for plant growth. The photo on the right here is another scanning electron microscope image showing biochar we collected from one of our field sites after it had been in the soil for about 3 years. So that means that this piece of biochar has had 3 years to get incorporated into all the ecological relationships that go on in soil. And you can see here the cell walls, and you can also see that there are grains of mineral soil stuck together on the outside of the biochar. And you also see these fine filaments, which are probably fungal hyphae. Biochar particles can become a hotspot for nutrients and moisture. which means that plant roots and microbes will seek out the biochar to access the nutrients and moisture that these organisms need for growth. As microbes and plant roots die, more and more organic matter accumulates on the edge of the biochar, which then also causes more particles of mineral soil to stick to the mix. So this lump of mineral particles, bits of organic matter, bacterial cells, and fungal hyphae is actually the beginning of a soil aggregate. And the photo shows an example of how biochar can stimulate soil aggregates to form. And aggregates are really important for soil health because they further help keep organic matter, nutrients, and moisture in the soil, providing a slow release supply of nutrients for plants. And this cartoon on the left now, this is a cartoon of a soil aggregate. It shows that the aggregate is composed of mineral particles, including silt, clay, and sand-sized particles, as well as bits of dead biomass or organic matter, shown in this darker brown. It also shows that plant roots here and fungal hyphae are growing around and into the aggregate, seeking out the nutrients that are contained in the aggregate. It also shows that the aggregates provide habitat for many types of soil organisms that all play a very important role in the soil food web. And that brings us to the third main category of soil characteristics, soil biological activity. So the cartoon on the previous page showed a soil aggregate providing habitat for a variety of soil organisms. And habitat is important for these organisms because it provides protection from their predators. The physical structure of biochar also provides habitats for microorganisms, even without being part of a soil aggregate. These photos here are more scanning electron microscope images from another researcher, Johannes Lehmann at Cornell, who's been a key leader in developing the biochar research field. The top photo shows fungal hyphae growing into the plant cells that we can still see visible in the biochar particles. The lower photo on the right shows other microbes, such as bacteria, that are colonizing the biochar pores. We can see that by these white arrows. Again, these pores in biochar are really the cells of the plant biomass that are still physically present. And as well as being attracted to the nutrients absorbed to biochar particles, microbes that colonize the pores are also more protected from their predators. And this can lead to changes in the diversity of the soil microbial community and changes in how quickly the microbes can cycle nutrients that are in the soil. And since soil microbes are the key organisms that decompose organic matter and cycle nutrients, benefits to the microbial community can lead back to increased nutrient availability for agriculture. But I think it's also important to point out that changes to the soil microbes is still one of the least understood areas of biochar research because microbial communities can be very complex and they also change naturally depending on soil moisture, temperature, and many other conditions. But these examples over the past few slides, I hope illustrate some of the ways that the physical, chemical, and biological characteristics of soil are continually interacting with each other as part of the soil ecosystem, and all of them can be influenced by biochar. So even though biochar is still a relatively young research field, we're beginning to accumulate enough published studies now to start studying the patterns and trends these studies are pointing to. So the two graphs on this slide are figures from a meta-analysis, which is basically a scientific study of the results from many other scientific studies. The horizontal axis on both graphs show the percent change in crop productivity relative to the control treatment used in an individual study. So the dashed red line here shows the 0% change. So 0% change would mean there was no difference in crop productivity for crops grown in biochar amended areas compared to areas with no biochar added. The graph on the left shows categories of soil pH on the right side of the panel. This ranges from soils with pH greater than 7.5 on the bottom to pH of less than 5 at the top. Now remember that pH 5 is relatively acidic for soils and is pretty typical for sandy northern forest soils. So the researchers looked at all studies of biochar amendments in soils with pH greater than 7.5, and they calculated how much biochar changed crop productivity compared to that study's no biochar control. And this is called the effect size, shown here in the blue dots. The wings on either side of the dot are error bars, and this shows how much variability there is around the average. Now, if the error bars overlap with the 0% line, we conclude that there's no statistically significant effect of biochar on crop productivity. So this graph shows that there's no statistically significant effect of biochar on crop productivity for soils when biochar was added to soils with pH between 6.5 to 7.5. In contrast, adding biochar to soils with pH greater than 7.5 does increase crop productivity by about 6%. For soils with pH between 5 to 6.5, biochar increases productivity by about 15%. And for the most acidic soils with pH less than 5, biochar increases crop productivity by 30%. That's quite a lot. And the grand mean here is the overall average, or like the average of all the other results. So the grand mean shows that there is an overall positive effect of biochar on crop productivity, even though if your soils are 6.5 to 7.5, you might actually see a negative effect. So we can interpret the graph on the right in the same way, except that the graph on the right shows, whoops, my slide is, a little stuck here. Okay, here we go. Oops. Got my laser pointer back. That's the graph on the right here. We can interpret in the same way, except that it shows the meta-analysis results of biochar studies for different types of soil textures. And this ranges from sandy soils on the bottom to clayey soils on their top. And this graph shows that biochar amendments increase crop productivity in all of these categories of soil texture. So they're all on the right side of the red line, and they're not crossing, the error bars are not crossing the line of no effect. The increase in loamy soils is pretty reliably around 8%, shown by these really small error bars, compared to the really large error bars around the average for clayey soils, which shows that the effects of biochar in clayey soils can be much more variable, even though it still shows a statistically significant increase in crop productivity. But what I think is important here is that the greatest increase in crop productivity again happens when biochar is added to sandy soils. And this creates an almost 30% increase in crop productivity compared to sandy soils without biochar. So both of these panels together are showing that biochar is most effective for improving crop productivity in soils that are acidic and sandy, exactly the types of soils we find across the Northern Lake States region. Okay, so the last few slides shows what we know about charcoal amendments and why they may have been used in ancient gardens. Now I want to talk a little bit more about the different ways that the charcoal may have been produced based on my background, both with biochar research and with studying natural charcoal materials that are produced in wildfires or prescribed fires. And we talked earlier about the pyrolysis and combustion conditions that happen during a fire. And I'm showing here, again, the photos that represent what I'll call the non-industrial situations that create char, ranging from cooking fires to a slash and burn approach to clearing land for agriculture, to intentionally starting fires to manage the landscape for berry production or hunting, or to building mounds that promote smoldering instead of flaming combustion. So all of these types of fires would create some amount of charcoal, but they would vary in how much char versus ash was produced. And they would also vary in how concentrated versus how spread around the char and ash residues were. So based on my background researching biochar and wildfire chars, this slide shows my hypotheses for what the characteristics and distribution of the charcoal in the ancient garden sites might tell us about how the charcoal was produced for the ancient gardens. Some of this information may already be known from the research Dr. Overstreet and others have already done. So I hope the way I'm framing it here can help tie together different types of information. So starting from the left, refuse middens. If the charcoal was just the leftovers or the waste from cooking fires, this would be a similar situation to the terra preta soils in the Amazon, which were basically the trash heap from the ancient village. In this case, I would expect that the charcoal would be mostly wood charcoal as the most likely type of cooking fire fuel. I would expect that its particle size and chemistry would be somewhat variable, which would represent a range of cool to hot fire temperatures and a mixture of pyrolysis and combustion reactions. I would expect that the charcoal amendments would be pretty localized, meaning there would have been repeated additions to the same site, such as a trash pit or a garden, that would accumulate gradually over a long period of time, years or decades. I would also expect that waste from cooking fires might contain animal bones or other artifacts, and this would be, again, very similar to the terra preta sites in the Amazon. If, on the other hand, the ancient garden sites were created from a slash and burn approach to clearing land, I would expect that the charcoal fragments would be from a mixture of plant species and plant parts, such as any combination of wood from conifers or deciduous trees, bark, pine cones, other seed structures, needles, leaves, and grass stems. This would also lead to a lot of variability in the size and chemical characteristics of the char. And also, clearing a site and burning the cut vegetation leads to less fuel for future burns. And this means that the first burn would likely create the most char, but later burns would just be burning up grasses and shrubs, or most likely just burning up crop residue if these sites had been used for agriculture. So the slash and burn approach would probably create a pretty even distribution of charcoal across a site, especially if it was intentionally mixed into the soil by these ancient gardeners. But based on the photos I've seen from Dr. Overstreet that shows the soil profiles at the garden sites, it seems that a slash and burn approach would probably not be able to produce as much charcoal as what I could see was present in the photos. So the slash and burn approach is pretty similar to the broadcast burn possibility. And the broadcast burn, these would also create charcoal from a mixture of plant species and plant parts. Basically, any vegetation that was there would be charred to some extent. This would result in char with variable size and chemistry in the charcoal pieces. And unless the fire was burning through a very dense forest, it would probably not produce a lot of charcoal and would probably be pretty patchy across the burned area. In this situation, the amount of charcoal produced would depend on how much and where the burnable fuels were located. Now, this photo for the broadcast burn that I have on this slide shows the modern broadcast burn in a red pine forest, but burns could also have been conducted in non-forested areas, such as a grassland or a pine barrens area. But again, the more times a site is burned, the less vegetation there will be for the next burn. So it's probably also unlikely that this type of burn would be able to create the amount of charcoal that's present at the ancient gardens. Okay, so the last photo shows a mound kiln. This is a way of deliberately creating charcoal. And this example from Kenya is used to create charcoal briquettes, which are then used as a cooking fuel. But this photo is just an example of a deliberate way to maximize charcoal production by controlling the amount of oxygen present during a fire. So for example, covering a fire after it has started, as long as it's not fully extinguished, would limit the amount of oxygen the fire can access. And limiting oxygen means you're limiting the flaming part of the combustion process, even though the pyrolysis stage can keep going. This means that a smoldering fire with little to no flaming lots of char left over and very little white ash. So it could be possible to create a fire in a trench or pit filled with wood or other types of fuel. And after it gets started a little bit, it could be covered but not fully smothered. It could be covered with wet vegetation or soil or even mixture. And I would expect that charcoal from this type of approach may be mostly wood char, which survives the charring process best and would also be easiest to haul around. I would also expect that the charcoal chemistry would be more consistent since the fire conditions would be more controlled and more consistent within this smoldering mound of fuel. And this approach would probably be able to produce pretty large amounts of charcoal in focused areas, which could then be spread around into a larger area. So from the little bit I know about the ancient garden sites from Dr. Overstreet and from my own background in biochar and wildfire chars, it seems that either the refuse middens possibility or the mound type approach to creating charcoal would be the most likely sources of charcoal, or at least to be able to produce as much as is present in the ancient gardens. But studying charcoal particles from these ancient garden sites will be important to be able to provide additional clues. So for example, examining charcoal fragments at high magnification can reveal what types of plant parts and even what species the char came from. This is possible from looking at wood anatomy, looking at the size, shape, and arrangement of plant cells, by looking at the characteristics of the stomata on the surface of the leaves. And these are structures that control the exchange of oxygen and carbon dioxide and even control moisture loss from plant tissues. It's also possible to identify species by the phytoliths present in the charred plant tissue or in the soil. And phytoliths are silica-based structures that plants produce inside their cell walls that can tell us about what species of plants have been present in the past. So the six images on the right are from research we've done in my group on wildfire chars. And the panels A, B, and C show scanning electron microscope images from charred pine bark, charred pine cones, this is jack pine in the middle, and then charred wood on the bottom. So these are on the right, the photos of what the SEM images on the left were taken of. So these images show us that charcoal characteristics can tell us about what type of plants and plant parts the char was produced from. The sizes and size class distribution of charcoal pieces can also give additional clues about how the charcoal was produced. These photos show charcoal pieces on the left, fragments in the middle, and particles on the right. And these are just different size classes of charcoal, bits of charcoal. So we know that charring makes wood more friable, which is more easily broken up into smaller pieces, and that the more extensively charred that the wood is, the more friable it is compared to less extensively charred wood. We also know that wood charcoal tends to break up into little chunks, but grass charcoal tends to be present more as flakes. Again, this physical structure of the char represents the physical structure of the original type of biomass. And this next slide here also shows that the elemental composition and the ratios between elements in char can also tell us even more about the temperatures used when the char was created. So this is one of the ways that the char chemistry, it gives us clues about how it was produced. So this slide is showing two Van Crevelen diagrams, and this is a standard way of presenting ratios between hydrogen and carbon atoms on the y-axis, the vertical axis here, and oxygen to carbon ratios here on the horizontal axis. The figure on the left is a study by, actually it's a typo by Liu, L-I-U, and others in 2013. This figure shows that the raw biomass for all this different species that these researchers studied is located in the right-hand corner of the graph. And this is showing that there's relatively high amounts of hydrogen and relatively high amounts of oxygen in the raw or uncharred biomass compared to the amount of carbon there. So there's still a lot of hydrogen and oxygen atoms present in this uncharred biomass. And remember that the charring process is pyrolysis, meaning breaking chemical bonds by heat. And this pyrolysis results in char that has a much greater concentration or density of carbon atoms compared to the original biomass. So looking at the left-hand corner of the graph, we're looking at the charred material that was produced at different temperatures. This is showing that the raw biomass, the raw biomass, which was then charred at 400 degrees Celsius, leads to a decrease in hydrogen to carbon and oxygen to carbon ratios compared to the original biomass. So the higher the charring temperatures, the lower we get in the hydrogen to carbon and the oxygen to carbon ratios. And we also see that the higher the charring temperature, the more similar the element ratios are across these different types of original biomass. So we can say that the variability in element ratios among types of biomass decreases as the charring temperature increases. We see the same pattern in the figure on the right. And this is a Van Crevelen diagram for wildfire chars, again showing that the uncharred biomass here contains much more hydrogen and oxygen compared to char produced from the same materials. Now the axes in this figure are not shown all the way down to 0. They stop at 0.6 on the y-axis and 0.3 on the x-axis. So these chars here would be most similar in terms of H to C and O to C ratios. They'd be most similar to about the 400 degree level in the left-hand figure. So this makes sense because in a natural fire situation, there's a mix of pyrolysis and combustion and variable amounts of oxygen present and not always very high temperatures. So we would expect natural chars to have more variable characteristics and to probably represent a somewhat moderate level of heating compared to very high levels of heating. So if we were able to identify the plant species from a piece of charcoal from the ancient gardens, we could then measure its atomic ratios in the lab and then either look into the published literature to find data like Liu and others presented, or do our own charring experiments in the lab to help determine the temperature that the ancient garden chars was likely produced in. And measuring atomic ratios on many pieces of the ancient garden char would then tell us about how consistent or how variable the char production temperatures were over time and in different garden sites. So the molecular composition of charcoal can also give us additional clues into its formation temperatures. And for this, we can investigate BPCA ratios. And BPCA stands for benzene polycarboxylic acids, which are molecular markers for charred biomass. Benzene is just another name for these 6 carbon ring structures or aromatic carbon rings that are concentrated together in charred biomass. And in the laboratory, we can digest a piece of charcoal in strong acid to break it apart into these clusters of aromatic rings. So break apart these big clusters into smaller benzene, smaller molecules. And each of these types of BPCAs tells us about how big the cluster of aromatic carbon rings were that it came from. And it tells us that by the number of carboxylic acid groups, the COOH groups, that are attached to it after it's separated from the cluster. So for example, B3CA comes from a smaller cluster of aromatic rings where it is less surrounded by other aromatic carbon rings. B3CA is shown here in blue. B6CA would indicate an aromatic ring that was completely surrounded. Oops, here we go. B6CA in green. It would indicate an aromatic ring that had been completely surrounded by other aromatic rings, shown here as the green, because it has 6 COOH groups attached to the benzene ring. Now, any piece of charcoal will have some areas with many aromatic rings condensed together and other areas that are not as condensed. So we're really interested in the ratios of these different BPCAs to each other and how much each BPCA contributes to total BPCAs measured. The figure on the right is showing the relative contribution or the percent composition of each of these different BPCA molecules to the total amount of BPCAs measured, shown as whatever total amount is measured is considered 100%. So, for example, for chars created at, on the horizontal axis here, I should point out, this is showing data from chars produced at different temperatures, ranging from 200 degrees to 1,000 degrees in the laboratory. So for chars created at 200 degrees Celsius, in this first bar here, we see about 30% of the total BPCA measures were measured as B3CA, and about 50% of the total measured was present as B4CA, compared to only about 10% each for both B5CA and B6CA. And again, B6CA comes from the most condensed areas, and B3CA comes from the least condensed areas. So if we look at the change in BPCA composition, moving from the low temperature chars to the higher temperature chars, we see that B3CA, the darkest color bar, decreases as charring temperature increases, and B6CA, the lightest color bar, increases as charring temperature increases. So if we also look at the white circles that are on top of each bar, these correspond to the axis on the right, and they show the total amount of BPCAs detected from each of the different temperature chars. So this shows us that low temperature chars contain a very small amount of total BPCA. But when we measure the BPCA composition, it's dominated by these smaller, less condensed forms. In contrast, chars formed at higher temperatures contain a much higher amount of total BPCA, and the BPCAs are dominated by these B6CAs, which are the molecular markers of higher temperature char. And these higher temperature chars have very low amounts of B3CA. So this research shows us that the total amount of BPCAs measured and the relative contributions of the different BPCA molecules can tell us about the temperatures that ancient charcoal was formed in. And this can again help us understand what the characteristics of the fires were that produced the chars and how consistent or variable the fire characteristics were at different dates or in different garden sites. So another important piece of the puzzle is how much char should be applied to soil. And if we're interested in knowing how much was originally added to these ancient garden sites, a first step would be to measure how much char still exists in the ancient gardens. So in general, the aromatic chemical structure that we see in char, shown by those polyaromatic carbon rings connected together, that chemical structure makes it pretty resistant, but not totally resistant to microbial decomposition. So this means that measuring how much char is in the gardens today should give a pretty good idea of the total amount that had been added over the time period the gardens were in use. So what was added should in general still be there. But there are a few different ways to measure total amounts of charcoal in soil, and sometimes they can produce slightly conflicting results. And this is a very active area in charcoal and biochar research today. So one approach is just to separate out as much of the visible char pieces as possible to find out how much mass of charcoal exists in a certain area of soil. Chemical analysis is another approach This typically involves a laboratory digestion to quantify charcoal based on its chemistry. And the BPCA analysis is one example of several types of lab methods that we can use. So for either of these approaches, once we get an estimate of char mass, we can then calculate the ratio of char mass to soil mass. So how much mass of charcoal exists per gram of soil. We can also calculate how much char mass exists per meter squared or per acre or any other unit of area. But I'm showing a question mark here in this puzzle because any of the measurements we do will only be an estimation of how these ancient garden amendments were created. Measurements we do now won't be able to tell us about how time and weathering have changed the ancient garden amendments since the sites were active. And it's important to remember that all chars, when they are fresh, have some amount of mineral ash or white ash mixed in. And the more oxygen present when the char was created, the more white ash will be present. This means that the residue from cooking fires or slash and burn type fires would probably have had a lot of white ash mixed in with the char when it was first added. And this white ash, remember, contains a lot of important nutrients like calcium, magnesium, potassium, and phosphorus. But it also tends to not persist very long, especially in sandy soils, because it can leach down physically into soil. And it can also, the nutrients in ash can also dissolve in rainwater, and then they're taken up and used by plants. Or they leach through the soil and out of the soil profile and are removed from the site. So this ash can be a really important source of nutrients, but typically lasts for a relatively short period of time. So over time, it's, again, that nutrients from the ash would either be used or lost through the soil. And especially the loss is high in sandy soils that are so porous. But I recently learned from Dr. Overstreet that there are areas in the ancient gardens that look like thick layers of ash that are still present. So this could happen if there's a lot of ash applied at once, because those the ash can, like charcoal, contribute to plugging some of the pores in mineral soil. So that would also help prevent the ash from leaching away. So what can current biochar knowledge tell us about how much char should be applied to agricultural soils? And unfortunately, this is a topic that is still not very well understood. And the simple answer is it depends. It depends on what the characteristics of the char are, what the characteristics of the soil are where you want to apply it, and what crops you want to grow there. And all of these factors influence how char influences soil properties and plant growth. So modern biochar gets a lot of attention for the positive effects it has had, but there are many examples where it has actually had negative effects on crop growth. The photo here on the left side of the slide is from a small demonstration project that we did using char produced from municipal solid waste. So this char was somewhat unique because it was produced from treated human solid waste, not from plant biomass. So it doesn't have that three-dimensional cell wall structures that plant biomass char tends to have. This char in this experiment also had very low carbon content, and instead it had very high ash content. The photo shows representative tomato plants that we grew from seed in the greenhouse in soils that were either had no biochar here in the pot on the left, to a low amendment level and to a high amendment level. And this roughly corresponds to about the 11 tons per acre and 33 tons per acre I showed in a previous slide. So this photo shows that this type of biochar had a negative effect on tomato growth and biochar. And the plants grown in the biochar treatments also showed sign of nutrient deficiencies. So it's important to remember that all biochars are not the same, and there can be a lot of variability in what the charcoal characteristics are based on what the char was produced from. The figure on the right shows another meta-analysis figure, and this shows the overall change, again, in crop productivity for different types of crops. Now, if we looked at the grand mean here in the middle, this shows that the overall result, if we lump all crop types together, is that biochar increases crop productivity by about 10%. And the effect is statistically significant. The error bars do not cross the zero line. But if we look at the results for individual crops, we see a lot of variability. And many of the crops have error bars that cross the zero line, which means there's no statistically significant effect of biochar on that crop's growth. In this meta-analysis, radish and soybean were the only crops that had statistically significant increases in productivity when they were grown with biochar. Maize or corn shows a trend towards increased productivity, but we can't say that the effect is statistically significant. Ryegrass clearly shows a negative effect, less productivity when biochar is added. So again, it's important to know what type of crop is intended to be grown with biochar, because that can really influence whether the crop responds positively or negatively. The meta-analysis by Liu and others also looked at changes in crop productivity based on how much biochar was added, and that's shown in this figure on the right. And the categories of biochar amount are shown here, with the amount shown in tons per acre in the first column, and metric tons per hectare shown here in the parentheses. And this study showed that the greatest increase in crop growth across all crop types and soil types resulted from the lower levels of char amendment. So there's a better response based on this meta-analysis when adding low or moderate amounts of char compared to very high amounts of char. And we don't have good research information yet about whether it's best to apply char all at once or gradually over time. But the example from the pteropodicytes shows that the pteropodicytes were formed gradually over time, with an average char content that's measured today of about 3.5 metric tons per hectare, which is on the very low end of the amendment levels that have been used in modern biochar research. Current interest in biochar has also moved away from thinking that applying biochar alone is the best idea. And now there's much more awareness that biochar is often most effective when it's combined with a nutrient source, such as compost. And from what I understand from Dr. Overstreet, the past research at the ancient garden sites shows that muck from riparian areas was also added to the ancient gardens. So this suggests that the people that created the ancient gardens actually already knew that mixing char with other types of biomass was the best approach. And this is something that the modern biochar community today has only recently begun to recognize. So I want to circle back to the original goal for this presentation, which was to focus on how biochar research techniques and knowledge can help understand these ancient garden sites that are part of the Menominee Nation. In the context of scientific research, our understanding of biochar is still pretty recent. But I hope that my presentation helped provide some insights into charcoal characteristics, how it was produced, how it is produced, what we know about how it can be used, and what we are still learning about how much to apply, as well as how its effects vary depending on soil type, biochar type, and type of crop. So there's still a lot to learn. But one of the questions Dr. Overstreet and I have discussed is whether the communities that established the ancient gardens were using biochar. And if we go by our modern definition of biochar, which includes charcoal-like materials that are deliberately produced and deliberately added to soil for management purpose, we can confidently say, yes, these ancient communities were using what we would today consider to be biochar. So this then raises some new questions for us today. For example, how are these ancient biochar amendments still influencing soil health and plant growth? Are the beneficial effects as long-lasting as what we see in the Amazon in the terra preta soils? And this then turns us back towards the future. Can the ancient gardens help identify what are the best combinations of biochar, soil types, and crops to use now to support efforts to increase food sovereignty and ecological stewardship. So even though Dr. Overstreet invited me to talk about what biochar research today can tell us about charcoal in ancient gardens from the past, I think studying the ancient gardens is really an opportunity to learn from the past to help create or to help restore a path toward a more sustainable future. And biochar amendments can help achieve this by helping move back towards a zero waste system where organic matter, including charcoal, is returned to the soil to support soil health. By supporting soil health, we can move toward a more sustainable future by supporting the soil's ability to provide the resources we need today without compromising the ability of future generations to meet their needs. And biochar may ultimately help move us toward a more carbon negative Earth system where carbon is stored in soil where it can support our planet's ability to sustain life. So I think this knowledge really goes hand in hand, both applying the tools of today to better understand the past and learning from the past to create a more sustainable future. These are some of the many people who have contributed to the information I've shared in this presentation and some of the many sources of funding that have made the work possible. I will show my references on the next slide if you're interested in additional information, on, especially my sources. And my contact information is listed on this slide here. So especially thank you to Dr. Overstreet and to Dr. Frank Kutka for helping make this possible. Miigwech, thank you for your attention and your time.